Assalamualaikum and good morning to everyone. Welcome to webinar Nutrition Research Priorities Series 4 2022. My name is Dr. Aswe bin Abdul Rashid. I'm the head of Nutrition Unit and MCRC at the Institute for Medical Research, National Institutes of Health, Malaysia, and I will be serving as your moderator today. In this 2 hours webinar session, you will hear 3 presentations from 3 speakers. They are Mr. Muhammad Naim Muhammad Nawi, Dr. Sofia Rashika Ismail, and Mr. Muhammad Azrul Azri Jamilan on interesting topics related to nutrients and non-nutrients analysis. They will also share their experience in securing research grants for their respective projects. Here are some of the housekeeping messages before the presentation begins. For all our viewers, please subscribe to NIH YouTube channel, follow our Facebook page, National Institute of Health Malaysia, share this live stream link with colleagues and friends. Questions will be answered after all speakers has given their presentations. Submit your question through Google Form link or the chat box. E-certificate will be provided at the end of this live stream after filling in the feedback form. Viewers may download NRP documents at the link provided for more information on nutrition research priorities. Before we invite the speakers to deliver their speech, I would like to invite the chairman of this webinar, Prof. Dr. Hamid Jan bin Jan Muhammad, to deliver his welcoming remarks and introduction of nutrition research priorities in Malaysia for the 12th Malaysia Plan 2021-2025. Prof. Dr. Hamid Jan works as a lecturer at the Nutrition Program, School of Health Sciences, University Science Malaysia since year 2007. He joined this institution immediately after completing his PhD at King's College London, United Kingdom. He holds a master's degree in nutrition from University Kebangsaan Malaysia and bachelor degree in nutrition and community health from University Putra Malaysia. He has successfully supervised 9 PhDs and 10 master's students, and currently he has 11 PhD students on various research topics. He works closely with the Ministry of Health Malaysia by being committee members of the Technical Working Group for Nutrition Guidelines, Children, Adults and Elderly, and other consultative activities. Dr. Hamid is currently the chairman of the Technical Working Group under Nutrition Research for the Ministry of Health Malaysia. Prof. Hamid also, his research interest is on maternal and child nutrition, obesity, vitamin D, and nutrigenomics. Let us hear the pre-recorded welcoming remarks from Prof. Dr. Hamid Jan.
Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to everyone. Alhamdulillah. Thank you very much, Dr. Aswir, for the kind introduction. And we are here today to learn, relearn on important aspects of nutrition. And it is a beautiful day today that we are able to collaborate together, bring together the Nutrition Division of Ministry of Health and the IMR, both under the same ministry, and both units are important in terms of the nutrition research in our country. And this webinar is specially designed so that we can learn from each other, we can share our knowledge to everyone, the young one, the senior person, the experienced and those who are not experienced, in terms of the direction of our nutrition research in our country. The NRP is a very important document. It is written together by experts in our country, particularly in the field of nutrition. And we all work together in trying to see what is the critical gaps in nutrition related knowledge and evidence. And by being able to highlight and identify this element, we will be able to suggest the direction of the nutrition research in our beloved country. And we also want to generate valuable information and evidence for policymakers to draw up and make informed decision on new policies. Because policies are important, evidence-based policies are much more important because it gives more strength to the policy, and the user will be more accepting those policies that is backed up by evidence. And whose responsibility is it to come up with the evidence? It is us, the researchers, the agency, the research institute, universities. We are responsible to come up with valid evidence, and those evidence can be used by several agencies, not only Ministry of Health, and this will be helping the policymakers to support their policy, whatever policy that they are planning to bring forward. And the third objectives of the NRP is to provide a platform for individuals or related organizations, uh, Ministry of Agriculture, Ministry of Health, Ministry of Education, universities, food and beverage industries, NGOs, everyone. We want to bring everyone into the platform because we want to highlight that nutrition is not only the responsibility of Ministry of Health. I think this is very important because nutrition comes from many spectrum, from the farm, from the agriculture, from the fertilizers, from the uh, irrigation to the education and move forward to the industry. And this whole food chain in terms of food production, the type of food that we are producing in this country and etc. is very important. So we need to have a platform, we can all sit together, we all can share our knowledge, our ideas, and we can also exchange our experience, and we learn from that. We learn from each other, and this platform is created. One of the objective is for communication between several stakeholders, and we want this to happen. We want this to be initiated as well so that we can build up the nutrition research in our country. As you all know, the NRP book is published. You can get it online in the PDF format on the Nutrition Division website. And just for those who are new about this, there are seven research priority areas in the NRP. We have done some webinars on maternal, infant, and young children. I think we have done on life course approach, nutrition deficiency and excess. And recently we did overweight and obesity, which is together with the NCD issues that related to obesity. And today we are here to learn more about nutrient and non-nutrient composition of food. And this NRP is designed, is crafted in line with the NPA-NM3, so that we are not, diverting from the national objective or national idea. Particularly for the webinar that we are attending today, which is nutrient and non-nutrient composition of food, it is linked with at least two of the NRP, which are two of the NPNM, which is sustaining food system to promote healthy diet and supporting efforts to promote food safety and quality. So this is how we ensure that the developed NRP is in line with the enabling strategies of NPA and M3. So, so we are here to see, and particularly when we talk about RPA7, nutrient and non-nutrient composition of food, there are several scopes that we can discuss. I'm not going to go through this in detail. I just want to indicate that the top four here is the main scope, and it indicates that one of it is to enhance nutrient data in combating nutrition-related diseases. We need a lot of data on macro, on micro, on phytonutrient, and etc. And it is very important data we need to develop so that we can relate all the dietary intake, and the more accurate information on dietary intake can be developed, and we could get a better and clearer picture when we link diet and disease. We also suggest a scope on to enrich nutrient data for ethnic, new emerging, and underutilized food. I think it is very important, okay, where there are emerging foods like hipsters, the boba tea, and etc. Several yogurt, ice cream. You can see many innovative food that we can see on our market, food market. We even like invasion of like international food, like food from Korea. Like if I go in Kota Baru nowadays, used to be people selling keropok leko along the street. Now there are more takoyakis, along the street, and it shows some transition of food that is promoted and sold to the community in our country. And we should not be left behind. We should explore this, we should investigate, and we should be able to have data on this so that we can put all into the building bricks of the data analysis, and then we could get a clearer picture. As I mentioned just now, phytonutrient is a very big word. It's a very important word, but we still do not have enough research on phytonutrients, and we need young people here or new researchers here to also consider doing research on phytochemicals, so that we could appreciate the plants that we have, local or non-local plants, and we could explore more, we could promote more on this plant. Ulam, for example, I think if you do more research on ulam, you may get something or some new information on the phytochemical and anti-nutrients or elements that it may have. So see, the science is evolving, and we should use all the latest technology that we have now to explore the plant and the food system that we have. And finally, one of the scope is to improve and develop food analysis method. Method is important. With new method, we could generate new knowledge, and that's why we have highlighted here that one of the scope over here is a method development. And here we are today together with the team from IMR. I again would like to give a big appreciation to Dr. Aswir Abdul Rashid, who is the head of unit in the nutrition unit IMR. And then I think without his effort, this webinar today may not be possible. But I think the whole team members of the IMR who are involved here, Mr. Muhammad Naim, Muhammad Nawi, Dr. Sofia Rashika Ismail, Mr. Muhammad Azirul Azri Jamilan, here volunteering their time and their expertise, sharing with us on three important topics that we will hear today. The first one will be exploring metabolism in normal and disease using metabolomics. Second one is exploration of lipid profiling and its advantages through lipidomics approach. And third one, miniaturization of vitamin D detection in food matrices and serum. These are three important topics, and it needs exploration and sharing with everyone. But I also hope that they are able to share not only their research topics area, but also their personal experience in terms of generating the idea of doing this research, managing the research in terms of grants and also resources. And this webinar, I believe, will be very beneficial to all of us who are interested in nutrient and non-nutrient analysis, and also new methodologies like metabolomics, which is very important, which can be applied in many areas of nutrition research in our country. So I think I would not want to speak a lot on this. I think the most important person are the speakers. You are the VIP today, and you have the responsibility to share. And it is I'm very happy that we are here today together, and we are able to learn from you all. And before I end, I would like to give a big appreciation to everyone who have registered and who are here with us in this webinar. I think your time will not be wasted. Inshallah, you will learn something new from this webinar, and there is some take-home messages that you will gain, and you can apply it in your current research or your future research. With that, I would like to end my opening speech with a thank you note. Thank you very much.
Thank you, Mr. Chairman, for the welcoming remarks. To those who are just joining our webinar, these are some of the housekeeping messages. Please subscribe to NIH YouTube channel, follow our Facebook page, National Institute of Health Malaysia, share this live stream link with colleagues and friends. Question will be answered after all speakers has given their presentations. Submit your question through Google Form link or the chat box. E-certificate will be provided at the end of this live stream after filling in the feedback form. Viewers may download NRP documents at the link provided for more information on nutrition research priorities. Now moving along to our session, please welcome Mr. Muhammad Naim Muhammad Nawi, who will be speaking on exploring metabolism in normal and disease state using metabolomics. Currently, Mr. Naim works as a researcher at Nutrition, Metabolism and Cardiovascular Research Center, Institute for Medical Research. He obtained his Bachelor of Science in Food Science and Technology from UITM and Master of Science from UPM respectively. He has a keen interest in the study of micronutrients and human-animal metabolomics. He is currently pursuing PhD in metabolomics at Taylor's University, Malaysia. Without further ado, Mr. Muhammad Naim Muhammad Nawi, the floor is yours.
Assalamualaikum and good morning to all. As part of the webinar Nutrition Research Priority Series 4, year of 2022, the title of my presentation today is Exploring Metabolism in Normal and Disease States using Metabolomics. First, I will talk a bit about NCDs. NCDs, which include CVD, diabetes, and chronic kidney disease, are the leading causes of mortality worldwide, linking to about 58% of the 57 million deaths in 2017. In Malaysia, NCDs were responsible for 74% of deaths in the year of 2019. CVD has the highest mortality in Malaysia. High blood sugar, high blood pressure, and high cholesterol are the major risk factors for CVD. As reported by the National Morbidity and Health Survey NHMS 2019, at least 3.4 million people in Malaysia currently live with two of these risk factors. According to a joint report by the World Health Organization and the Ministry of Health Malaysia on the impact of NCD and their risk factors on Malaysia's gross domestic product, Malaysia's economic burden of management of NCD is estimated at around 179 billion ringgit in the year of 2020. The Global Burden of Disease Study, or GBDS, measure health loss from diseases, injuries, and risk factors to improve health system and leads to the introduction of Disability Adjusted Life Year, or what we call as DALY. DALY is a matrix for the countrywide assessment for the likelihood of NCD's developments. These risk factors can be divided by three distinct categories, which is behavior, metabolic abnormalities, environmental, as well as occupational. And according to this figure, dietary risk is the top risk factors in Malaysia. As we can see, behavior risk from poor food consumption choices, such as consumption of fast foods, processed foods, and sugar-sweetened beverages, increases the risk of NCDs. Abnormal nutrient metabolism in chronic disease states. The macronutrient, lipid, protein, and carbohydrate provide energy and essential components to sustain life. The abnormal metabolism of these micronutrients, such as carbohydrate, lipid, and protein, involve oxidative stress, which is associated with inflammation and insulin resistance, which eventually contribute to the development and pathogenesis of all of these NCDs. Carbohydrates are a micronutrient that contributes energy to the body, and excessive intake has been linked to subsequent weight gain and obesity. Phosphatidylcholines, a class of phospholipids, has also been linked to metabolic syndrome, adipose tissue problem, as well as inflammation. Lastly, suboptimal intake of protein will lead to malnutrition in chronic kidney disease patients. What is metabolomics? Metabolomics is an evolving field of life science that utilize advanced analytical chemistry techniques with sophisticated statistical methods to comprehensively characterize the metabolome. Phenotype refers to the observable traits and is determined by factors listed here such as genetic and epigenetic factors, age, environment, nutrition, and lifestyle. Metabolomics is basically the study of metabolites produced within cell and biologic systems as a byproduct of metabolism, and it is able to measure changes as it happens. Next, we will talk about multi-omics. Multi-omics is basically the integration of various omics to explore systems biology. Genes at the start code for proteins, which are used by cells to transport and process metabolites, which in turn are used to assemble the very genes and proteins that help make these metabolites. This intimate connectivity means that a single base change in a protein coding gene can potentially lead to a 10,000 change in the concentrations of specific metabolites. This amplification phenomenon in going from DNA to proteins to metabolites is also seen in terms of the number of genes, which is around 20,000, versus the number of expected metabolites, which is around 1 million, and the diversity of chemicals used to assemble genes for bases or proteins, which is around 20 amino acids, versus the diversity of chemicals seen in the metabolome, which is about more than 3,000 chemical classes. Due to all of this, the size and complexity of the metabolome make metabolomics a particularly difficult branch of omics science. Nutri-metabolomics is a term referring to the merging of metabolomics and nutrition research. Diets on its own are complex, highly variable between populations and ethnicities, and there is limited understanding of food chemical compositions, food-specific biomarkers, and personalized nature of the human metabolism itself. The application of metabolomics to nutrition provides an intersectional physiological evaluation of dietary metabolites as a cause-effect of nutrition interventions or trial. Metabolomic techniques have been applied to understand the pathophysiological generation and uptake of metabolites as a consequence of chronic disease processes on human metabolism. I'll list down a few examples of metabolite patterns of nutrients in chronic disease. Sugar metabolites, including glucose, dihexose, mannose, arabinose, and fructose, are being examined for associations with the prevalence of diabetes, with results being unclear. Metabolomics approaches have also been incorporated in studies looking at the incidence of CKD, CKD progression, and the hemodialysis population. In terms of metabolic syndrome, there are studies looking at the increase of amino acids such as alanine, leucine, and isoleucine. Metabolomic approaches can be either targeted or non-targeted. Non-targeted metabolomics approaches involve a global profiling of the metabolome. This approach is typically employed in hypothesis-generating studies such as biomarker discovery, where comprehensive metabolite identification is generally not the ultimate goal. On the other hand, targeted metabolomics refers to the quantitative measurement of a select group of metabolites, examples such as amino acids, lipids, sugars, or fatty acids, in order to investigate specific metabolic pathways or to validate biomarkers identified using non-targeted metabolic profiling. Thus, non-targeted metabolomics often provides more information than targeted metabolomics. but targeted metabolomics typically is more quantitative. Various analytical techniques have been used, but nuclear magnetic resonance and mass spectrometry are the most common analytical tools in metabolomics research. The high reproducibility of NMR-based techniques and the high sensitivity and selectivity of MS-based techniques mean that these tools are superior over analytical techniques. NMR is normally used for untargeted metabolomics, while MS-based techniques are used for targeted metabolomics. This is a typical summary of metabolomics workflow. Blood is most commonly used biological metrics in metabolomics. Blood collection tube selection is indeed important. Do we use EDTA? Do we use lithium heparin? Or do we use normal serum tube? These are some of the things to consider. Having said that, there are also studies that utilize urine, saliva, and tissue. Data acquisition very much dependent on the choice of analytical platform, either NMR or MS. Normally, after data acquisition, we usually need to do some sort of data cleaning, which can include steps such as normalization, log transformation, scaling, and peak alignment. Then we will proceed with statistical analysis, which could be either univariate or multivariate. Some of the analysis that we could use includes principal component analysis, PCA, PLSDA or PLSTA. PCA normally we use to look for overall patterns of our data, while PLSDA and PLSTA can be used to help see whether any specific metabolites contributed to the separation and variation between the control and treatment groups. The workflow will then be concluded by biomarker discovery stage, which is made up but not limited to feature identification, pathway analysis, and neural network. What do we do when we want to design a metabolomic study? 3 key questions to ponder. Normally, at the starting point, we need to consider the suitability of either a targeted or untargeted approach. Do we plan to do it in humans or animals, because this will have further implications down the field later? How do we plan to design the study? Composition of cases, composition of controls. Do we need positive or negative controls? Next is the biological matrix of choice. Which one do we use? Do we want to use serum? Do we want to use plasma? Do we want to collect urine instead? Or the pathways of interest are best observed through tissue samples instead? This is important because each biological matrix have different collection protocols that we need to observe. Is it doable? Do we need to preserve our tissues using liquid nitrogen? We need to consider what are the potential problems ahead when we discover our choice of biological matrix. The most important of all, the analytical platform, which is more suitable for your analysis. Do we plan to use NMR? What frequency you want to use? 400 MHz, 500 MHz, or 600 MHz? Is it a targeted or untargeted or a combination of both? Do you want to use MS? Will you require a special protocol for sample preparation? Each on its own have its own strengths and weaknesses. Do we need a special equipment which we don't have? Will that be a problem in the future? Those are some of the things that you need to consider. At the end of the day, you need to work with what you have and adapt accordingly. Now I will talk on my own experience applying for a grant for my Metabolomics project. I will just focus on the few important questions during the session. First, I choose an appropriate design that is suitable for my study. I'm doing an animal study, which I'm using Zucker rats to study the effects of micronutrients permutation on progression of diabetes, obesity, and atherogenicity. I'll have to explain to the panel why this model is suitable for what I want to study. So at the end of the day, I need to be up to date with my literature. Normally, I will just look at previous trends, current trends, as well as future trends in my respective fields. Then the panel will ask me about the design of the groupings. How many is the number of positive controls, the number of negative controls? Do I have enough number of rats? Is it enough? What will be my backup plan if any of it died during the course of the experiment? Is my sample size calculation correct? Then I do need to provide the sample size calculation during the defense stage. And at the end of the day, one of the most important thing in animal work is logistic and costing. So I need to provide a comprehensive plan and timetable of planned procedure. I need to show that I have prepared for everything And the panel will ask for justification, why I need to buy this item, why I need to buy that item. So we will need to show to the panel that those items are necessary for our research and we have to adapt accordingly. So if you do everything properly, then normally the project will be considered for funding approval. If not, then we will be asked to revise, make the necessary adjustments, and attempt to defend the project again. So that very much concludes the grant application sharing experience from me. So we are approaching towards the end of my talk, and I would like to talk a bit on the challenges that I face in conducting this study. I divided it into 3 parts, which is before, after animal arrival, and the endpoint of experiment. I only listed some of the major ones here. Firstly, in terms of the experiment itself, I've never done any previous work on NMR, so this is a totally new thing for me. Me and my colleagues spent 8 months just optimizing our protocols and methods. It was a huge challenge to set up a brand new protocol for this study. We have been blessed and fortunate to receive help and assistance from various local collaborators to do this. If this is not possible, it will definitely affect the overall execution of the project. Other than that, before the arrival of the animals, I need to put up with delays from the logistic side, the breeder side of the rats itself, and all other things. And not to forget COVID, the movement control order, MCO. A lot of things are outside of my control and it can get really frustrating at times, but I'm glad we managed to solve all of the issues. So once the animal arrive, we just need to make sure the execution of the project goes smoothly. We need to do daily observation, take care of the rats, and make sure we complete the treatment duration. And lastly, the endpoint. It's more towards study coordination. We need to plan for blood protocol collection, euthanization procedure. Do we use carbon dioxide? Do we use isoflurane? Should we consider using ketamine injection instead? Will it have any effect on metabolites later? Then we need to get the correct blood tubes. Do we want to use EDTA? Do we want to use heparin? Should we buy the right volume, either 4 ml, 6 ml, or 10 ml? Normally for animal work, we will be using 4 ml instead of 6 or 10. We need to also consider long-term storage for samples. Is it minus 20? Is it minus 80? We also eventually need to plan for data processing and all. So at the end of the day, we need to make sure we have all of the necessary tools to finish our experiment. In summary, what is next in the field of metabolomics and nutrition? Today's ultra-sensitive instruments provide analytical scientists an unprecedented opportunity to detect more compounds at lower concentrations, resulting in a larger and larger proportion of unknown compounds to be identified, most of which are not found in available existing databases. This is particularly true in the field of nutritional metabolomics, where the focus is not only on human endogenous metabolites, but also on food-derived compounds introduced with the diet. And finally, the future evolution of nutritional metabolomics lies in integrating with other evolving omics techniques and with other well-defined tools used in disease diagnostic. So that concludes my presentation. Do write your questions and I will try to answer it during the Q&A session. I hope my presentation has been beneficial and you will have some idea what research you can do through metabolomics in regards to nutrition. If you need further discussion, you can definitely email me and I'll get back to you whenever I can.
Thank you very much, Mr. Muhammad Naim, for presenting the interesting topics on metabolomics. He also mentioned about the differences in the types of omics available by category, and he touched on the application of metabolomics in nutrition research settings. In addition, he also touched a little bit on the process for applying the research grant under Ministry of Health. Now we move on to our next speaker, Dr. Sofia Rashika Ismail. She will be sharing her experience on the exploration of lipids profiling and its advantages through lipidomics approach. Currently, Dr. Sofia Rashika is a researcher and medical officer at the Nutrition, Metabolism and Cardiovascular Research Center Institute for Medical Research. She graduated with a medical degree from the International Medical University, Malaysia, and pursued MSc in Public Health at London School of Hygiene and Tropical Medicine, United Kingdom. She is completing her PhD in Community Health at the University Kebangsaan Malaysia. Her area of interest include cardiovascular epidemiology, cardiovascular genetic epidemiology, lipidomics, and evidence synthesis. In 2017, Dr. Sophia won the Merdeka Award grant for international attachment for her research on lipidomics, profiling, and risk of early onset myocardial infarction. The grant allowed her to learn and conduct her research at the University of Cambridge. So let's invite Dr. Sophia Rashika Ismail to deliver her speech. Dr. Sophia Rashika, the floor is yours.
Assalamualaikum warahmatullahi wabarakatuh. A very good morning. Today I'll be presenting on the exploration of lipid profiling and its advantages through lipidomics approach. So lipidomics is a fairly young discipline, of which its establishment came about in the early 2000s. With growing interest in this field, by 2014, there has been a surge in research trends aimed at analyzing lipids in a more holistic way, especially focusing on medicine and nutrition. So what exactly is lipidomics? Lipidomics is considered part of metabolomics. Lipidomics is known as the discipline to obtain integral information of all lipids in the biological system concerning cellular signals, membrane architecture, transcriptional and translational modulation, cell-cell and cell-protein interactions, and response to environmental changes with time. In other words, it's the understanding of lipids in all of their cellular roles in a biological system. So how does lipidomics differ from metabolomics? Well, essentially, both methods study the characteristics and interactions of metabolites with similar workflows. However, lipidomics studies only hydrophobic nonpolar compounds of lipid species, whereas hydrophilic polar compounds such as amino acids, carbohydrates, and nucleic acids are uniquely studied in metabolomics. So Lipid Maps is an online platform that provides access to lipid nomenclature, databases, tools, protocols, and other relevant resources pertaining to lipidomics. As there are hundreds of lipid species in a biological sample, Lipid Maps provides a standardized classification and nomenclature of these lipids. Based on lipid maps, lipids are categorized to 8 major classes. These are fatty acids, glycerolipids, glycerophospholipids, sphingolipids, sterols, phenols, saccharolipids, and also polyketides. So similarly to metabolomics, there can be both targeted and untargeted approaches. In targeted lipidomics, there is previous knowledge of the lipids involved in the biological processes under study, which allows for the usage of the most appropriate analytical platform. For this approach, LC-GC-MS is usually coupled to a mass detector of high sensitivity and selectivity such as triple quadruple. Sample preparations can also be catered specifically to clean up the desired lipids. In untargeted lipidomics, it detects a wide range of lipids that can be achieved by using LC-GC or capillary electrophoresis or even NMR. Here, the sample prep is generally simpler to achieve the widest possible lipid coverage. In lipidomics, the shotgun approach is beneficial for fast and sensitive quantitative analysis of lipid species. This approach explores the lipid properties through direct infusion coupled with MS platform. So essentially, the lipidomics workflow involves sample preparation, data acquisition through analytical platforms, and data analysis. Sample preparation is determined by the sample type, lipids to be studied, as well as the analytical approach. The selection of appropriate solvents is the most critical factor for efficient extraction of lipids. A mixture of polar and non-polar solvents are precisely selected to extract lipids efficiently. Each extraction method has its advantages and disadvantages, therefore, sample characteristics need to be understood beforehand to ensure adequate solvent penetration and extraction of lipids. The most commonly used methods for human blood plasma or even serum samples are the Foch and MTBE methods. Although there is evidence of various analytical platforms used in lipidomics, LC-GC-MS and NMR are the most commonly used platforms. Data processing and data analysis of lipidomics data are also similar to the protocols in metabolomics. In healthy states, lipidomics can describe specific lipids involved in dynamic physiological changes. Additionally, it can provide insight into lipid control in cellular biology, such as in the structural integrity of cellular membranes, in signal transduction, and so on. Evaluation of lipidomes associated with disease states will also increase understanding of the metabolic processes relating to the disease. Studies in individuals with metabolic syndrome, cardiovascular diseases, and others have highlighted several prominent lipidomes. Lipids change in every cell and body fluid, particularly in response to food intake. Lipidomics has also shown to play a role in personalized nutrition. Although quite similar to the concept of precision nutrition and sometimes interchangeable, precision nutrition refers to a conceptual framework covering a wider set of individual features, allowing an effective and dynamic nutritional approach. While personalized nutrition based on genes is already being implemented successfully in numerous studies, precision nutrition may still lack sufficient evidence for full implementation given its complexity. The close relationship between the nutrigenomics and metabolomics or lipidomics is seen in this slide. Ideally, our healthy plate recommendations would include all the different variables in the precision nutrition plate. So lipidomics approach can also be used for identification of nutritional biomarkers. In identifying biomarkers of exposure, native molecular forms which are otherwise not observed in other methods of analysis may be explored. In identifying biomarkers of effects, nutritional compliance with treatments may also be observed. And lastly, lipidomes may be used as biomarkers of health or disease with known and unique biomarkers identified in metabolic syndrome, cardiovascular diseases, and other common diseases. In the subsequent slides, I'll be going through my previous work and current work on lipidomics. My work essentially evolves around coronary heart disease, specifically on individuals with first myocardial infarction. So I've performed and I'm performing lipidomic studies on samples from the Maverick study. So Maverick was a multi-center, nationwide, retrospective case control study of genetic lipid and other risk factors in early onset MI in Malaysia. We were able to recruit 2,547 MI cases and 2,500 MI-free controls from 17 centers throughout the country. We included 83-item FFQ as well as blood samples and also history and anthropometry measurements. My first work in lipidomics was during my attachment at the University of Cambridge. The attachment was made possible by the Merdeka Award Grant for International Attachment. This grant is open to all Malaysians between the ages of 22 to 35 years to engage in short-term collaborative project for up to 3 months at a chosen international institution. My traveling, stipend, and all research-related costs, including transfer of samples, purchase of consumables, and reagents, were all covered by the grant. The grant applications are open every 2 years, with the next opening to be in Q1 of next year, so do keep a lookout. During my attachment at the Core Metabolomics and Lipidomics Laboratory, Cambridge, I had learned on the sample prep methods, procedures, data acquisition, and data processing, as well as basic statistical analysis of the data. Although there was still a lot to be learned on the different steps of the lipidomics workflow, the attachment provided a very good stepping stone in the understanding of the method and in establishing the method in our lab here in IMR. A shotgun approach of approximately 600 samples from the Maverick study was performed. Our data demonstrated significant distinction in 9 out of 13 major lipid classes between individuals with and without MI. So after coming back from the attachment, I wanted to implement the methods in IMR. There were, of course, several advantages and disadvantages of the method learned in Cambridge. First of all, it was a high throughput method and robust established method for lipidomics in cardiovascular diseases. Due to the direct infusion techniques, we were able to acquire data for 384 samples within 23 hours. Thus, this will allow for a shorter duration of sample analysis for the entire Maverick samples. However, the direct infusion method required the purchase of Advion Nanomate. That alone costed R500,000. And thus, after going through the NIH research community, the grant application was rejected due to the high cost of accessory wanted to be purchased and its lack of technical support in Malaysia. Subsequently, we sat down to write a new proposal for a method that would be cost-effective, adaptable, and optimizes existing facilities, yet robust and reproducible. This led to a successful second grant application under the NIH. As the lipidomics workflow is not rigid and can be customizable according to the type of sample, type of lipid species being studied, and analytical platforms, we made several changes to the sample prep and data acquisition methods. Although it took a bit more time to establish a robust and reproducible method at our center, it is definitely a lot more cost-effective and has the potential to be used in other diseases as well. So to end my presentation for today, the understanding of metabolic functions and dysregulation of lipids through lipidomics approach has its benefits. Lipidomics in nutritional research has huge potential, but further exploration is needed in Malaysia. Thank you.
Thank you very much, Dr. Sofia Rashika, for delivering the presentation on the potential of studies using the lipidomics approach and the possibility to combine nutrition research on what's so-called nutrigenomics, nutri-lipidomics. Before we invite the third presenter, I would like to inform the audience once again on for some of the housekeeping messages. For all our viewers, please subscribe to NIH YouTube channel, follow our Facebook page, National Institute of Health Malaysia, share this live stream link with colleagues and friends. Questions will be answered after all speakers has given their presentations. Submit your question through Google Form link or the chat box. E-certificate will be provided at the end of this live stream after filling in the feedback form. Viewers can download NRP documents at the link provided for more information on nutrition research priorities. Okay, let's move to our third speaker, Mr. Muhammad Azrul Azri Jamilan. Mr. Azrul will deliver his talk on miniaturization of vitamin D's detection in food matrices and serum. Just like the two speakers, Mr. Azrul is also a researcher at Nutrition, Metabolism and Cardiovascular Research Center, Institute for Medical Research. He obtained his bachelor degree in analytical chemistry from USM and MSc degree from University Putra Malaysia. In 2019, he won a gold award at Unimas Innovation Technology Expo. Without further ado, let us invite Mr. Muhammad Azrul Azri to deliver his speech. Mr. Muhammad Azrul Azri, the floor is yours.
Assalamualaikum everyone. Today I will share our experience of handling research project with the title of Miniaturization of Vitamin D Detection in Food Matrices and Serum. First, let me introduce the overview of vitamin D's in general. Vitamin D is part of fat-soluble vitamins that consist of vitamin A, D, E, and K. which this vitamin is non-polar compound and it is important for proper functioning of human metabolism. As for vitamin D's group, it consists of vitamin D2 or ergocalciferol and D3 cholecalciferol. These two types of vitamin D's only differs with the existing of double bond and a metal group as shown in the chemical structure. In our body, the consumption of vitamin D will further hydroxylated into several vitamin D metabolites, including 25-OHD, 1,25-OH2D, and also 24,25-OH2D. The vitamin D level can affect cancer, cardiovascular disease, hypertension, depression, and immunity. The deficiency of vitamin D may lead to bone diseases such as rickets in children and osteomalacia in adults, and also to a non-bone related disease. On the other hand, the excess of vitamin D through diet could cause a hypercalcemia or vitamin D toxicity. According to the table shown here, the recommended range of 25-OHD in serum must be in between 20 to 50 nanogram per ml. There are several sources of vitamin D, where 90% come from the exposure of human skin to the UVB radiation in sunlight, which in turn it will produce vitamin D3 or cholecalciferol. This type of vitamin can also be obtained by the consumption of food such as salmon, cod, and milk, and fortified food to be such as infant formula, cooking oils, and also from the food supplements. The absorption of vitamin D2 can also be obtained by the consumption of plant-based foods such as fungi and mushroom, and from food supplements. The absorbed vitamin Ds are carried by a protein in blood called vitamin D binding proteins. This is due to the insolubility of vitamin D in bloods, which later on transported to the liver to be converted into a non-active metabolite called 25-OHD or calcidiol through hydroxylation. Calcidiol is the most abundant metabolite in serum and plasma, thus making the detection of vitamin D represented by the concentration of calcidiols. Whenever needed by the body, calcidiol will be transported to the kidney to be further hydroxylated into an active form of vitamin D metabolites, which is known as 1,25-OH2D, or in short, we call it as calcitriol. These forms of active metabolites used to regulate calcium and phosphorus intake for bone mineralization. Next, we go to the method of detection that is commonly used to detect 25-OHD, where the most common one is the use of high-performance liquid chromatography with UV visible detector, or known as HPLC UVVs. HPLC UVVs often used as a reference method due to the reliability and reproducibility of the methods. However, the instrument have several drawbacks, such as it uses a large sample volume of at least 500 microliters, and it also requires a sample preparation procedure before analysis. A more sophisticated and high-end instrument of LCMS, or we call it as high-performance liquid chromatography coupled with mass spectrometry, is also used by other researchers to detect calcidiol due to the excellence of LOD and sensitivity of the instrument, and it uses very low sample volume of several microliters. The drawback is that the instrument is very costly and high maintenance cost. It also requires sample preparation similar to when using HPLC UVVs. These methods are known as a direct detection without using any labeling substrate. A more affordable and reliable method is more preferable in daily diagnostic laboratory. This includes the use of spectrophotometry detection, such as UV detection in ELISA and fluorescent detection in chemiluminescence assay, and also the radioactive detection in radioimmunoassay RIA, although it is becoming uncommon nowadays. Typical approach of this method of detection is by using a labeling reagent attached to the 25-OHD, such as using HRP or horseradish peroxide and also alkaline phosphate in using ELISA, and also using labeling of luminol or isoluminol when using chemiluminescence detection, which give off light emission to be detected by the fluorescent detector, and also using radioactive element such as iodine-125, which is detected by the radioisotope detector in RIA. A fixed amount of labeled 25 OHD typically introduced in the respective system during the reaction that will compete with the 25 OHD presence in the samples. These two 25 OHD labeled and unlabeled will bind itself competitively to the anti-25 OHD antibody, which in turn, the respective signal is inversely proportional to the 25 OHD concentration in the samples. This technique is called competitive immunoassay binding. So we go to the introduction of electrochemical biosensor. The diagram here shows a portable version of electrochemical sensor device that is attached to the screen printed carbon electrode. In our study, the use of electrochemical biosensor is the main approach of our method of detection. The development of detection method using this technique is quite new and considered as a novel approach. But before going any further, let me explain the brief definition of the term. The term biosensor means a method to observe biomolecules interaction of target compounds by any physical or chemical changes using a sensor device. Sensor is a device that observe changes to a measurable value, whereas electrochemical biosensor is a sensor that converts any current changes observed in biomolecule interaction of target compounds occurred on an electrode into a signal that can be read. Electrode is often used as a surface for biomolecule interaction with the target compound. Modification of electrode allow the versatility and biocompatibility of target compounds to be detected. The combination of using aptamer and electrochemical biosensor further enhance the novelty of our research. The combination called aptamer-based biosensor, or in short is known as APTA sensor, will be used instead of using anti-25 OHD antibody, so that the aptamer can selectively capture 25 OHD. Aptamer is a RNA or single-stranded DNA sequences and can be designed via a method, in vitro method, that is known as SELEX, or we call it as systematic evolution of ligands by exponential enrichment. Aptamer can also be prepared by software prediction, which is known as in silico approach. Aptamer can be reused, thermally stable, does not easily denatured compared to antibody. The design of aptamer can be customized based on the required binding affinity, making it more versatile to be used in the detection methods. Let's see the difference in vitamin D interaction when using ELISA and using APTA sensor. In ELISA, once the antibody of 25-OHD is immobilized to the plate, mixture of known amount of biotin labeled 25-OHD and unlabeled 25-OHD from the samples will compete to bind to the free sites of antibody, where higher concentration of 25-OHD from the samples will lead to a lower binding of biotin labeled 25-OHD. The binding of HRP to the labeled 25-OHD occurs when streptavidin labeled HRP is introduced, which the existence of HRP on the surface cause the oxidation of TMB as a chromogen substrate to change color, enable the detection using UV detection. On the other hand, APTA sensor, the immobilization of aptamer on the electrode surface allows not only 25-OHD but also vitamin D2 and D3 to be captured, which the concentration of this vitamin D is directly observed by the oxidation peak produced by a redox indicator that is known as ferrocyanide. Higher concentration of captured vitamin D will give off a lower signal due to the rate of diffusion of redox reaction decreases. The oxidation is found to be inversely proportional to the vitamin D concentration. The ability of capturing both vitamin D and its metabolites of 25-OHD enables the application of detection in serum as well as in food matrices. So the advantage of using sensor, including shorter total runtime, which typically takes around 30 minutes, and it is more sensitive due to the advantage of using electrochemical detection. The method can be further developed into a portable device in the future, and also the sample volume required is small, typically from 5 to 10 microliter. Our study falls under D1 category in research priority area 7, which is the improvement of analytical methods and related methodologies. Our research was funded by the Ministry of Health Malaysia under Program Penyelidikan Biomedical IMR. The procedure of applying this grant is through the submission of research proposal to the JPP IMR. An MRR registration is a must. Here, list of collaborators inside or outside IMR must also register to the system. The research protocol must be uploaded. Grant application and MREC application have to be applied through the system, although our research was exempted from the MREC application because the study does not involve any human-related samples. When everything is done, the proposal presentation will be done to the appointed panels in JPP NIH. After that, some revision might be needed based on the feedback we get, and the last step, that is the last step before starting the research project. Here are the list of challenges during the study. The use of 25-OHD 25-HD has a smaller size of around 0.4 kilodalton. So it is very hard to determine the binding affinity of the aptamer and the vitamin D. Also, the small molecule caused the difficulty for us to differentiate the signal produced with or without the vitamin D. This problem has been overcome by us by using the derivatizing agent to increase its size. Secondly, vitamin D is a fat-soluble vitamin, which means it must be dissolved in organic solvent. Due to this, it is hard to control the sample incubation on the electrode as it was easily dispersed. Thus, only small volume of sample will be used for the incubation, and mixing of organic solvent with water can also be used to improve the solution the sample solution surface tension. Our future plan is to transfer the newly developed aptamer-based biosensor to a portable device for miniaturization purpose. We also plan to further the study to the next phase, which is to validate the miniaturized device with real-life samples of human serum and also other types of food matrices. Ultimately, we want to commercialize the validated miniaturized device as an assay kit for the point-of-care testing device, and also for an affordable, easy-to-use, and reliable routine for food analysis detection. So that's all for me. Thank you.
Thank you very much to Mr. Muhammad Azra Azri for presenting the interesting topic on vitamin D and the several possible ways of determining vitamin D in samples. He also touched a little bit on the grant application process. Okay, we have completed the presentation session, and now before we start with the Q&A session, I would like to remind all the attendees to fill in the feedback form. You can just scan the QR code in your live chat box. We will have 2-minute breaks before we start with the Q&A session. Stay tuned with us. Okay, welcome back everyone. Now let's move to the Q&A session. Let's see if we have any question from the audience. Okay, we have a question from Siti. She is asking to Mr. Muhammad Naim. The question is about the sample prep. Is sample prep will be the same for biological samples? Mr. Muhammad Naim, can you answer that question, please? Thank you.
So thank you, Siti. Your question is that you want to know whether that the sample preparation for the biological sample will be the same. So in terms of plasma and serum, normally the preparation is the same. Basically, we will take around 200 microliter of that sample and mix with a buffer. And normally this buffer is made up of potassium dihydrogen phosphate with the addition of TSP as the internal standard, as well as imidazole as the pH indicator. However, for urine, there's a slight difference because for urine, normally in order for us to prevent the microbial growth, we will add around 0.03% of sodium azide. So this is done in order to preserve the urine samples and prevent the microbial growth for the long-term storage of the samples. Having said that, in terms of tissue samples, it's entirely different. In terms of tissue samples, normally it will require a homogenizer of some sorts. You have to homogenize the samples first, and then normally you have to dry the samples and then re-dissolve it in the same buffer that I have mentioned earlier. So that is basically the summary of all the sample preparation for different biological matrices. Back to you, moderator.
Thank you, Mr. Muhammad Naim. I hope that answer your question, Ms. Siti. We got another question to Dr. Sofia Rashika from Noor. She would like to know why we choose MS platform instead of NMR for lipidomics analysis. And once Dr. Sofia Rashika has answered that question, maybe Mr. Naim can add some more to give the answer. Thank you.
So thank you, No, for the question. So in general, in lipidomics, MS is considered as the standard and preferred platform due to its sensitivity and specificity. In general, in lipidomics, chromatographic separation is essential to allow for lipid classes to be quantified separately based on the number of carbons, double bonds, and also other characteristics. So for this reason, the usual platform that's used is LCMS, and of which I have used for my current study. This separation is not possible with NMR, thus NMR was not considered. However, in shotgun lipidomics, whereby there is direct infusion ESI, direct infusion electrospray ionization, this allows this is coupled with MS, which allows for quantification of lipids without separation, without prior separation. The nano ESI actually provides a very stable ionization environment, and it enhances the intensity of the signals, thereby giving more very robust robust quantification of the lipids, and also with this method usually very minimal sample volume is used. Having said that, although there are actually various platforms that can be used to analyze lipidomics, metabolomics, at the end of the day, it is based on your available facilities, the type of sample that you are analyzing, and volume of sample, of course, and also the types of lipids that you are analyzing. I think maybe Encik Naim can elaborate more on why he chose NMR for his method.
So thank you, Dr. Sofia, for that answer. So in addition to the question, so I will just add a little bit more on why I choose NMR. As Dr. Sofia has mentioned, an MS have its own advantages in terms of separation and everything else. NMR normally does not provide separation, it works differently. Having said that, NMR is much less sensitive than MS, that is a fact. And for me, I always feel that the inclusion of multi-platform is the best way forward. But the thing is, sometimes due to constraints such as costs and financial, normally you have to work with what you have. So in this case, we use NMR because it's the best and cheapest option that available for us at the time. However, going forward, I feel that inclusion of multi-platform is the best way for you to extend the coverage of metabolites and lipidomes. So that is basically the answer for that question. So back to you, moderator.
Thank you, Mr. Muhammad Naim and also Dr. Sofia. Okay, we got another question from Yana to Mr. Muhammad Ezra Azri. She would like to know about the cost of estimation for producing a portable base detection. Mr. Muhammad Ezra, can you answer that question, please? Thank you.
Okay, thank you, Miss Yana, for the question. Actually, the portable electrochemical sensor itself costs around 8,000 to 10,000, and it is actually also depending on the supplier, what type of brand of the sensor that you are using, and in our case is actually 8,000 to 10,000, and including the aptamer, the cost of aptamer, the cost of buying reagents, chemicals and all the consumables, the sum total of all of this as a kit should be less than 20,000. And also the running analysis of per analysis of the samples actually is much cheaper. It can actually cost not more than 30 ringgit from my estimation. So as a whole, as a POCT device in the future, this device, as a kit, the cost can be further reduced when produced in a large scale. Usually, it's like that. So back to the moderator.
Okay, thank you, Mr. Muhammad Ezra. But there's got another question also to you, Mr. Ezra, from Rihanna. Because you mentioned about the aptamer that can be prepared via in silico or in vitro. So what type of aptamer preparation
regarding what type of preparation of the aptamer that we are using in our current study, actually we have used both type of aptamer, which first one we tried to do aptamer that is prepared via in silico technique, which is based on the software prediction. However, the result that we get is not that good. So we also tried the other one, the aptamer that is prepared using in vitro, and luckily we got the result that we want. So in our case, we prefer to use the aptamer that is prepared in vitro. Using in silico is okay for me. It can be done, but maybe further optimization for choosing what the design for the arrangement of the aptamer must be further. More development must be done first before we tested it on the real in a wet lab. So in our case, we just ordered the in vitro aptamer that has been published previously by other research team, but in future maybe we can proceed with to prepare ourselves. So that's all.
It seems that there are also a question on the aptamer from UCSI University to Azirul. How can this aptamer-based biosensor be used using other sample types, for example, antigen?
Actually, aptamer can be applied to many type of targets, and is actually depending on what is the sequence of the DNA that you want to design. And actually, based on the SELECT method, in vitro method, that method is extracting the high binding affinity of the aptamer towards the the target that you want. So in this case, if you are using antigen, so based on that, they can actually select the aptamer. So it can be applied in biosensor by immobilizing the aptamer on the sensor surface. So we can actually functionalize the aptamer either with using streptavidin biotin approach, or also COH, or COH with NH3 binding as a covalent bonding. So that's how we immobilize the aptamer on the electrode surface as a biosensor detection.
Thank you, Mr. Muhammad Azrul, for that answer. I hope that it answered the question. We got a question also to Dr. Sofia Rashika from Nurul. Could you elaborate on give examples of using lipidomics for nutritional biomarkers of effects?
Okay, thank you, Nurul, for the question. So generally, nutritional biomarkers of effects is related to a given function or a biological response related to a single nutrient or even interactions of several nutrients. A known example would be a comprehensive lipidomics analysis on omega-3 fatty acid that was used in the treatment in DHA-based treatment, multivitamin treatment for pediatric NASH non-alcoholic steatohepatitis patients. Yeah. So, then back to you, moderator.
Thank you, Dr. Sofia, for your answering that questions. We got a question to Mr. Muhammad Naim from Sue, from IMR. How are we to differentiate exogenous and endogenous metabolites? Can you give the answer, Mr. Muhammad Naim?
So thank you, Sue, for that question. Basically, metabolites are divided into two distinct categories. One is endogenous and one is exogenous. So endogenous metabolites are naturally produced by an organism, while exogenous is normally not produced by organism. Having said that, depending on the groups of those metabolites, it could be a difficult task to differentiate it unless those exogenous metabolites comes from drugs or toxins which are not overlapping with the endogenous metabolites. Having said that, there are research areas that are focusing towards the separation of endogenous and exogenous metabolites. One example of this area of research is food intake biomarker. As example, research has been done to show that the presence of proline and betaline as a biomarker for citrus consumption could be used in order to differentiate consumption and non-consumption. So basically, that is an ongoing area of research. There is still much to explore. So that is basically my answer to that question. Back to you, moderator.
Thank you, Mr. Muhammad Naim. We got a question from UPM to Mr. Muhammad Azri. Is it possible to use immunoassay-based reaction to be applied on the electrochemical detection?
Okay, regarding the question, immunoassay-based detection, that means you have to use antibody to be immobilized, immobilized on the electrode. So in this case, it's actually possible, and however, the sensor that you are using should be the antibody itself will cause the instability of the sensor. So that means the reproducibility of the electrode must be not good from batch to batch of preparation. So that's why we prefer to use aptamer instead of antibody.
Thank you, Mr. Muhammad Azrul, for answering that question. Back to you, Mr. Muhammad Naim. This is a question that in my mind, because just now you in your slide presentation, you mentioned about the biological matrix that can be used for metabolomic study. But you didn't put whether it is possible for feces determination using for metabolomics for metabolites?
Let me get this correct, that you would like to know whether it's possible for us to use feces as our biological sample. I personally haven't used any feces samples. Having said that, I've known people and colleagues from other departments and other research groups who have utilized feces, and normally the preparation is very much the same as tissues. Having said that, I am not really familiar with the details behind it, but the concept behind it is similar. Normally, you have to homogenize the feces samples first, then you have to dry it under vacuum, and then you have to choose the appropriate solvent for you to redissolve it. Then normally, we will use the same buffer that we use for other samples before we inject it into NMR. So I hope that answers your question.
Thank you, Mr. Muhammad Haim. Because I'm also have something in my mind, because sometimes people also sweating, sweat. The metabolites can also come from the sweat. So do you have any comment on that?
In terms of sweat, do you mean that in terms of metabolites, can we analyze metabolites from sweat? Is that your question?
Yes.
Okay, basically, we can analyze anything. It's just that in terms of sweat, I am not sure exactly how are we going to collect that. But in theory, yeah, no issue. We can use sweat to look for metabolites using NMR. As long as we have a way to collect it, we have a way to analyze it. No issue.
Okay. Thank you, Mr. Muhammad Naim. Okay, back to you, Dr. Sofia Rashika, because you are the recipient for the Merdeka Award grant, and I think you are the first Merdeka Award grant recipient in IMR, is it?
Yes.
Can you share with us the process of applying that grant, and maybe it's very others can also follow the same way. Yeah.
Thank you for your question, Mr. Moderator. So I think like I've mentioned in my presentation today, so the Merdeka Award grant for international attachment is a good opportunity for young researchers up to the age of 35 to embark into a 3-month attachment at a chosen institution. Previously it was a 1-year, previously the applications were open every year, but currently it is open only every 2 years. The next one will be early next year. So if you are eligible in terms of age and interested in having a fully paid attachment of 3 months at your chosen institution, please do have a look at the website early next year on the deadlines and the application dates. With the application, the most important document and the most important thing about the application is your proposal. You would have to submit a 2-page proposal with the application that would describe concisely and clearly your intended research during the 3 months attachment. I think it's important to describe and elaborate comprehensively the intended outcomes, not just at the end of the attachment, but also mid-term and long-term outcomes that would be beneficial to the Malaysian population. This is important because the grant is open to different fields. And then if you look into the history of grant recipients, I think majority of the winners have been doing research on environmental factors. I think mainly because it's the benefits, the potential benefits, has been clearly explained in those cases. Having said that, even though if it's a 2-page proposal, it should not be very technically scientific. Because the Merdeka Award committee members who will be reading through the applications are from various backgrounds. They can be from the scientific fraternity, but they can also be philanthropists and also individuals from the corporate organizations, especially their sponsors, Shell and Petronas. So it has to be a proposal that can be easily understood by anyone from various fields, yet still be technically sound and with big potential impact. So my advice is to to look into what has been done, what can be done, and think big on your potential benefits to the Malaysian population. Other than that, I think it's the usual requirements for any grant applications. You need to submit a budget, which there has been no limit, but it has to be justified. So then again, think big, look out for the application dates next year, early next year, and it will definitely be an experience worth going through. Okay. So back to you, moderator.
Because with that travel grant, you travel to Oxford, Cambridge University to learn about their lipidomic technique, isn't it? So can you explain a little bit about your experience while you were there?
Okay. All right. So I chose lipidomics for its potential in myocardial infarction for various reasons. And then even though I have been interested in lipidomics for some time, I have had no experience in doing lab work. I am a medical doctor and trained doctor, so I'm not familiar with doing lab work, playing around with instruments, especially MS. So that opportunity provided by the grant, and then to be able to work with one of the best labs in the world for lipidomics was truly enriching. They guided me from step one, from the basics on how to handle samples, how to prepare the samples, to using different instruments, and then up to data analysis. Even with the current study, they are still collaborators and guiding me through my journey. I know the 3 months attachment was not a very long attachment, but it was a very good boost in knowledge and experience for my future plans in lipidomics.
Thank you very much for your explanation. Okay, I got also a question to Mr. Muhammad Azirul about the mushroom. When you mentioned about mushroom in your slide presentation, and as you all know, mushroom is a food, or is a functional food that contains high vitamin D. So is there any particular reason why mushroom got high vitamin D content? Maybe because it can easily synthesize in their, something related to the sunlight. Can you comment on that?
Okay, thank you, Mr. Moderator. Actually, I'm not the expert of this kind of samples, I mean, particularly in mushroom, but I can comment maybe because the mushroom itself, generally we know the mushroom itself, by exposing of sunlight, is actually can naturally produce the vitamin D, but in the form of vitamin D2, or we call it ergocalciferol. So that's why the mushroom contains high in vitamin D, but it's actually in the form of vitamin D2. Although it's almost similar, it only differs by a metal group and a double bond compared to the vitamin D3. However, there are some reports actually stating that the biologically activity of vitamin D2 form of vitamins is less compared to the consumption of vitamin D3. That's what I know, but more than that, I don't know how to explain further.
Okay, thank you, Mr. Muhammad Azri. Okay, is there any other question? Let's see if you have got a question from the audience. If it's not, I think we can. Okay, we can. If there's just no other question, then thank you once again to Mr. Muhammad Naim, Dr. Sofia Rashika, and Mr. Muhammad Azrul for answering those questions and for the great presentations. On behalf of our Technical Working Group on Nutrition Research, Ministry of Health, and IMR, as well as NIH as a co-organizer, it was a great pleasure to have all of you with us. So this concludes the webinar series for 2022. Thank you to all the attendees for supporting this webinar. We hope that you have learned and enjoyed this webinar. Till we meet again at our next scheduled NRP webinar. And then before we end the webinar, we'll show the corporate video of NIH. Thank you to all and have a nice day ahead. Assalamualaikum and bye-bye.
The Institute of Kuala Lumpur was established in 1900 by the Resident General of British Malaya, Sir Frank Swettenham. Its aim was to promote the health status of the local population and to explore the great and unknown field of tropical medicine. The institute was renamed as the Institute for Medical Research, or IMR, a year later. Initially, the focus of this research was on beriberi and malaria. Subsequently, it goes expanded into the causes, treatment, and prevention of a variety of tropical diseases such as cholera, smallpox, leprosy, tuberculosis, typhoid, filariasis, dengue, melioidosis, scrub typhus, and nutrient deficiencies. IMR was formerly located at Jalan Pahang, Kuala Lumpur, before moving to Shah Alam recently in a new research complex together with other research institutes under the Ministry of Health Malaysia. This specially designed building is enhanced and equipped with advanced and high-technology machines and instruments. It is now recognized to be a high-impact center in biomedical research at national and global level, with the presence of Biosafety Level 3 or BSL-3, Arthropod Containment Lab or ACL, Good Laboratory Practice GLP, and Biomedical Research Core Facility BioRCF. Upon independence, IMR became the research arm for the Ministry of Health Malaysia, where research conducted has been aligned with the country's research priorities. In more recent times, research at the IMR has progressed in gathering in-depth knowledge of allergy and autoimmune diseases, primary immunodeficiency diseases, and the development of allergy test kits. The IMR has developed its expertise in nutritional biochemistry, where there is active involvement in preclinical studies on metabolic syndrome, diabetes, cardiovascular health, and endocrine-related diseases. The Institute's iodine test kit is used nationwide for monitoring purposes under the Iodine Deficiency Program. The dawn of the next millennium encountered many issues that were related to contaminants and pollutants in the environment and in the workplace, along with their impact on human health. Thus, in a forward-thinking manner, IMR conducted vulnerability assessment on impacts of climate change on health and health delivery. Cancer research at the IMR has vigorously looked at diagnostics, prevention and early detection, as well as identification of novel biomarkers and targeted therapy for precision medicine. For this, the Institute has been equipped with a specific pathogen-free or SPF animal facility and biobank. The Institute has established a unique interactive e-database called Global Information Hub on Integrative Medicine, or more commonly known as GlobenMed, which strives to become a referral source on traditional and complementary medicine, T&CM. In addition, herbal research activities are focused on the evaluation of herbal extracts under certified GLP conditions, assessing efficacy of herbal products using in vivo as well as in vitro systems, phytochemical analysis, as well as structure elucidation. The IMR has continued to provide in-depth research related to infectious diseases, especially during outbreaks, and it is the reference center for COVID-19, Zika, dengue, influenza, leptospirosis, melioidosis, and malaria. Epidemiological research activities have helped to establish the distribution of diseases, its factors, and how it helps in controlling diseases. The IMR provides specialized diagnostic tests nationwide related to organ and blood stem cells transplants, diagnoses of inherited metabolic diseases, inborn errors of metabolism, IEM, genetic diseases, and multiple myeloma. Over the years, the IMR has emerged and been recognized for excellent research achievements. IMR is the leading center conducting Wolbachia research in Malaysia. A research team from IMR made a breakthrough in wound healing with the use of maggots. It is a type of biotherapy using live, sterile fly larvae or maggots in non-healing wounds to remove dead cells and reduce bacterial contamination of the wound. The efforts achieved the ultimate goal of primary health care by the World Health Organization. IMR is also the reference research institute in early outbreak case detection, such as COVID-19 in Malaysia, particularly during the pandemic. The institute has equipped itself with a BSL-3 laboratory and an enhanced capacity building and vaccine development as a preparedness plan for any possible future outbreak. In the international arena, the IMR serves as World Health Organization, or WHO, regional and collaborating center for infectious and tropical diseases under various programs. The IMR has produced a few patented products ranging from insecticides, dengue, chikungunya, and many more, from which some of the patented products have been commercialized. The century-old experience in research has emerged the Institute to continually illustrate excellent service with new challenges and high pursuit as the IMR transforms its biomedical facility to support research both nationally as well as internationally. IMR is to be the world-renowned institution for biomedical research.